Hello, everybody, and welcome. Thank you so much for joining us. This is a webinar provided by the U.S. HUPO's ECR committee and is a part of an ongoing initiative where we're trying to work with our friends up with the Canadian Proteomics and combine to bring some really cool interacting and interesting informational webinars to our membership and everybody else who logs on. So my name is Amanda Smithers. I'm a member of the U.S. HUPO ECR committee. I am a postdoc at Harvard Medical School.
Hi, I'm also Amanda, and I'm a postdoc at Genentech now, and I'm also on the US HUPO ECR committee, and I was previously on the CNPN committee as well, because I did my grad school in Vancouver, so I'm really excited for this collaboration, and glad we can bring the two groups together.
So today, our webinar is going to feature two fantastic researchers, Dr. Thomas Kisslinker and Dr. Wilhelm Haas. And our overall format will be each presenter is going to have about 20 minutes. They're going to give some really great talks on the theme of cancer proteomics. And then we're going to spend the next 20 minutes in a combined kind of Q&A. And so that gives us an opportunity to kind of connect the different information together, as well as gives our awesome attendees plenty of opportunities to pitch any questions you may have. So you'll see in the Zoom box, there is a spot for Q&A. At any time, please go ahead and submit those, and we'll just keep them all together for the Q&A session at the end of the presentations. With that, I'd like to go ahead and introduce our first speaker, Dr. Thomas Kisslinker. So he is a professor in medical biophysics at the University of Toronto. And today, Dr. Kisslinker will report on N-glycoproteomics from patient-derived xenograft models for the discovery of novel serum biomarkers for high-grade serous ovarian cancers. Extension of this platform to 7 different tumor types is going to be aimed at the discovery of novel cancer-specific tumor surface markers. And I'm super, super excited for this presentation. So with that, I'll give it to you.
Thanks, Amanda. Thanks for the nice introduction, and thanks for having me. So let me see if I can still know how Zoom works. And get my laser pointer. And okay, great. Hold on, let me get this little square out of the way. All right. All right, so what I thought I'm going to do here, especially since it's a talk for the early career researchers, or at least organized by the early career researchers, is that I give you a bit of an overview of what the lab does. So rather than giving you some super polished talk of something that's published, you don't really need me for that. I'll start with something that's published and then show you kind of work that's actually ongoing right now. And so let's see if I can move on. Yeah, here I can. So my lab has been using patient-derived xenograft models in combination with glycoproteomics for the discovery of serum-based biomarkers for several years now. And I'll show you first published work from a previous graduate student where we applied the strategy to high-grade serous ovarian cancer models. I'll show you how we detect human-derived peptides in the serum of these PDX models, and then use these data to systematically develop PRM assays that could potentially be used as biomarkers for the detection of ovarian cancers that mimic CA-125, which is the current biomarker for ovarian cancer. I'll show you how we use this data and validate it in 2 small serum cohorts from patients. And then what I really want to spend most of my time on is how we use this data and expand it to an additional cohort of human patients, and most importantly, how we expand the strategy to additional PDX models, 7 different tumor types. So first, I want to give a little bit of a background on ovarian cancer, since this is the tumor types we initially worked. Ovarian cancers is the most common and most lethal gynecologic malignancy in Canada, as well as in the US and the world. It exists in 5 different histological subtypes. You can see this here on the right. And each of these can be further subdivided in multiple molecular subtypes. And this is based on data from TCGA or from CPTAC. High-grade serous ovarian cancer is the most common and the most aggressive subtype. And this is the subtype that we are particularly interested in. Most of these high-grade serous ovarian cancers 75% at an advanced stage. So this is this means that these cancers are kind of silent early on, and when the cancer presents, it's almost kind of too late already for these patients. High-grade serous ovarian cancer have an interesting initiation and progression. So most of these tumors initiate either at the fallopian tube epithelium or at the ovarian surface epithelium. And then very early in the initiation and progression of this disease, small tumor clusters or spheroids release from this primary site and colonize distant sites within the abdominal cavity, mainly the liver or the omentum. So that suggests that early detection of this disease is extremely challenging. So keep that in mind. So here's how these cancers are treated. And that treatment really has not changed much in the last decades. So it starts with very aggressive debulking surgery, and then is followed by chemotherapy. And these chemotherapies are usually carboplatin or Taxol-based chemotherapies. And then following surgery, the disease, the efficacy of this treatment and disease progression is monitored by CA-125. So this is a glycoprotein using an FDA-approved ELISA assay. Most of these patients recur within 2 years. The reason for that is most likely incomplete debulking. So these thousands of tumor foci at presentation, they cannot all be surgically removed, or some other reasons like drug-tolerant persisters. And as a result of this, survival of high-grade serous ovarian cancer is very poor. Most of these patients have a 5-year survival of 10 to 30%. And then here on the right-hand side, you can kind of see a CA-125 longitudinal expression in serum. So that means 75 to 80% of these patients present with high values of CA-125 in blood. This goes down following surgery, goes further down during chemotherapy. And then patients then enter remission. And eventually this disease comes back, CA-125 rises again, these tumors become resistant to second and third line treatment, and these women unfortunately pass away from their disease. So we are interested now in using the strategy of glycoproteomics and patient-derived xenografts to find tumor markers that have longitudinal expression profiles that mimic CA-125, but we're particularly interested in kind of this stage. So can we have markers that potentially detect minimal residual disease better than CA-125? So CA-125 not that good in that stage, or potentially detect recurrence earlier. And that would give you more lead time to test out different second and third line treatments, which then potentially could improve outcome in these patients. So this is what we decided for the initial project overview, or for the project that we designed. It was done by a former PhD student, Ankit. So we kind of made use of very large PDX strategies that are ongoing at Princess Margaret Cancer Hospital. So we kind of piggybacked on work from Ben Neal's and Laurie Ailes' lab, published here on PDXs of high-grade serous ovarian cancer. But we kind of redid certain things. So we kind of took primary tumors and we IP engrafted them into these immunocompromised NSG animals. And that gives you a completely different take rate and tumor growth kinetics compared to these initial studies where these tumors were mammary fat pad engrafted. At endpoint, we surgically resect tumors, collect serum from these engrafted animals, and also collect serum from these unengrafted animals. And then we go through this kind of somewhat complicated workflow. So very early right at the beginning of this workflow, we spike a processing control. So this is a yeast glycoprotein invertase, gene MSAC2. We extract and digest these proteins to peptides, and then we use hydrocyte chemistry to specifically enrich for glycosylated peptides through this kind of what you can see here, oxidation and coupling of these hydrocyte beads to these generated aldehydes. And then because we're using PNGSF to release these glycopeptides from these beads, we're effectively only looking at N-glycopeptides. These N-glycopeptides are then analyzed by classic shotgun proteomics, and it goes through various kind of analysis strategies that I'll show you a bit in the next couple slides. So here's some of the reasons why we're interested in N-glycoproteins. As you probably know, most classically secreted or surface markers are N-glycosylated. These proteins are more stable in blood than non-glycosylated peptides. These bulky glycans protect them from proteolytic digestion. And then maybe most importantly, virtually all FDA-approved cancer protein biomarkers are glycoproteins, like C125 that I just showed you for ovarian cancer, or PSA for prostate cancer. So here's results. So in total, out of these 12 different PDX models, we detected around 4,000 or so glycopeptides. So this is at the peptide level. And then here you can see in these 3 different sample types that we analyzed, so tumor tissue, the engrafted serum, and the unengrafted serum, how they split. And then, of course, for the context of discovery of human-derived glycopeptides, probably these 649, or this quadrant where engrafted serum and tissue overlaps would be the most interesting. So if you're really ambitious or maybe a little bit silly like we were, then you can also try to develop PRM assays from these tumor unique glycoproteins. Turns out that's maybe not so good. Show you that later. And then if you just perform annotation analysis of this entire set of proteins, you can see, as expected, these proteins basically exclusively map to either classically secreted or membrane proteins, with the vast majority, probably 98 or 99% of the glycosides in the ectodomains of these membrane proteins. So that kind of intuitively makes sense and validates the strategy. And then probably the most interesting component of these PDX models is that your tumor tissues are effectively mixed species tissues, where your tumor that is engrafted is derived from a human being, and your stroma at the time of surgical resection of that tumor in the mouse is basically derived from the mouse, so from the host. So that means if you're detecting bioinformatically, or by mass spec and then by bioinformatic analysis, a human glycopeptide in the serum of these animals, by definition, it had to derive from the engrafted tumor. And here you can see some of these data. So here we detected approximately 1,600 or so glycopeptides in the serum of these animals. And here we ranked them by intensity of the peptide as a proxy of abundance of that parent protein. And then in these stacked bar charts here on the bottom, you see them split in septiles. So for example, the 230 most abundant and so on peptides. We assign species to it. So I don't have full time to go into how we assign species, but there's all kinds of thoughts that goes into this. And you can see in the most abundant peptide, as you would expect, the vast majority of these serum proteins are derived from the host, so from mouse. As you get lower in abundance, more and more human unique peptides are detectable. And that intuitively, again, makes sense, right? A protein that's secreted from a tumor kind of needs to make it into blood and then needs to be detectable by a mass spec. So you're probably expecting somewhat lower abundance proteins to be derived from that engrafted tumor tissue. On the other hand, if you look at the solid PDX tumor and you again do that species assignment, you don't really see this association of species type with abundance of that peptide. That again kind of intuitively makes sense. You wouldn't necessarily expect that a tissue that contains mouse or human proteins, there's any kind of bias in that species in that solid tumor tissue. And then I really go quickly over this, what we do for this project, so I get to the unpublished data. So I'll just show you some flowcharts here to summarize this. So now we take these peptides from these glycopeptides from these 2 quadrants of the Venn diagram. We assign species, so this is human unique and human unique peptides. We apply certain biophysical and annotation criteria that would hopefully help us when we design these PRM assays. So you can see there's around 386 that pass these strategies. We synthesize all of them as stable isotope labeled unpurified peptides, go through various rounds of tweaking of these assays. So there's unfortunately some methionines in these peptides that can be oxidized, brings the number of assays up. And then there's some peptides that just don't have very good signal to noise, so we kick them out early. So that leaves us with 450 unique targets that we're following. And that means 450 heavy and 450 light targets that we need to follow. So we then build an assay, a scheduled PRM assay for 900 targets in the study via parallel reaction monitoring mass spec. And then here's that first kind of round of verifications using longitudinal patient-derived serum samples. So you can see we have longitudinal serum samples from high-grade serous ovarian cancer patients at these different kind of stages of disease. So we take these 450 assays, kind of develop them. Just as a little side note, if you're building an assay for 450 PRM targets scheduled, probably not assays absolutely optimized. So keep that in mind. Then we apply these assays to two small cohorts, 10 patients, 50 samples in total. All of these have matching serum CA125 data from the FDA-approved ELISA assay for CA125. And here you can see that's actually what this ELISA data looks like. As I said, goes down through treatment, kind of stays low in remission, and then rises again in recurrence. So in the first cohort, we apply all these assays and we mine it for longitudinal expression profiles that look like this. So we actually ask, do some of these markers already from chemo complete in the remission time point start to rise and then further rise as that cancer recurs? 27 of these targets show these profiles. Now we buy them as highly purified aqua peptides, tweak that PRM assay a bit further and apply it to an additional cohort of 10 high-grade serous ovarian cancer patient samples for markers still show this kind of expression profile. As you can see, there's all kinds of heterogeneity in these kind of samples. And here's the 4 markers that we detected normalized to the diagnosis time point. You can see nicely they go down following surgery, further go down as a result of chemo. And then here our markers start to rise already in the remission time point, which C125, at least in this cohort, doesn't. So to summarize this part, so we have shown that the combination of PDX models and chemoproteomics enables us the detection of human-derived peptides, either in tumor tissues or in serum. And we can use this data to systematically develop PRM assays that we can then apply to longitudinally collected patient serum samples. So now a postdoc in the lab, Debbie, who's an analytical chemist, they're probably the only ones that you can convince doing these kind of studies, took these 4 markers, optimized these PRM assays, and applies them to an additional cohort. A graduate student in the lab, Minusha, was very interested in this particular PDX strategy. And I'll show you, she expanded this to 7 additional tumor types. And Minusha is particularly interested in looking actually at the tumor compartment of these PDX models. She is in particularly interested in human-derived membrane proteins for either molecular biology and cell models or for the development of antibodies that eventually could be used for immunotherapy development for ADCs or CAR T cells. Show you some data in the next couple of minutes. So here's the cohort that Debbie had in hand. And, you know, getting these cohorts, even in a large cancer center like Princess Margaret, is actually not that easy. So it's still a relatively small cohort. So we have 34 patients and around 6.5 or so samples per patient, or 200 time points per patient, so 212 samples. You can see here the annotation for these individual color points. And here for the first time, I actually also put you the actual time axis on it. So you can see recurrence occurs at completely different times in these patients. So then Debbie monitored these 4 markers that I showed you. She also developed a PRM for a C125 glycopeptide. That PRM is nowhere near as good as the FDA-approved ELISA. Then we also have 2 peptides from the processing spike-in from our yeast protein. So Debbie optimized all kinds of settings, how much serum you use, what beads you use, what's the bead to serum ratio. She optimized and shortened liquid chromatography gradient, optimized all kinds of scan settings on the PRM, and then again evaluated, do these markers in this cohort have this kind of a tumor load profile? Again, with the idea, do they rise early? So that's ultimately the goal that we set out to verify. So yeah, just show you this kind of summarized. So I show you this in two things, really focus mainly on the heat map on the bottom. So first we ask, do these markers as a result of treatment drop? So do they go from diagnosis, following surgery, following chemo, do these markers drop? And we just effectively mine these data by slopes. So if it's a negative slope, goes down, we give it a here purple color. If it's not, nothing happens, it's gray. And if it rises, it's red. So you can see CA-125 does actually quite good. Our markers, they also don't do so bad. There's some kind of heterogeneity in there, of course. Not every marker in every patient works. And then LTBP1 is probably the marker that most closely reflects what CA-125 shows you. And the harder question is, following end of chemotherapy, is there a rise in the markers? And here we ask, is there just a general rise? So you can see CA-125 does really good here, but I just want to tell you, right, this really only means it rises from here to the end of this last time point. CA-125 is usually flat here. And here's our other markers. Again, LTBP1 kind of looks nicely compared to CA-125. And here, if you zoom in on some patients with a little bit more granularity, so here you can see a subset of these patients where we have multiple time points in remission. Again, we normalize all this data to the diagnosis time point. So you can see CA-125 drops following treatment, kind of stays flat, and then finally comes up again in recurrence. LTBP1 mimics this, has maybe a little bit of a bump after surgery, also doesn't maybe have as much of dynamic range, but you can clearly see that it slowly starts to rise, likely earlier than CA-125. Of course, still needs validation additional cohorts. And here I show you one of these patients blotted out. You can see this may be nicer. So CA-125 drops nicely, so it certainly has most likely a better dynamic range. Stays flat and then really only rises once these patients recur, either based on rising CA-125 or based on imaging, actually. And LTBP1 kind of drops down to the end of chemo and then quickly starts to rise. So probably has slightly better lead time in detecting recurrence, at least based on this cohort compared to CA-125. Okay, and then finally, I want to show you some work from these additional PDX models. So now this is Minusha, a grad student in the lab's work. And she kind of, again, piggybacked on to what Ankit already generated for these high-grade serous ovarian cancer models. So she now has a total of 85 unique PDX models, so from 85 unique patients. There's multiple different tumors. So we have, for example, engraftment replicates here. So that's why the number goes higher. She collects the tumor tissues, the serum tissues, and then again, sera, and then she also has from unengrafted animals controls. What's interesting here now, so we have these 7 different tumor types: GBM, head and neck, lung squamous, lung adeno, pancreatic, colorectal, and the high-grade serous. We also compare different engraftment sites. So as I told you earlier, the high-grade serous, they're IP engrafted. These here are the typical subcutaneous. Then our GBM models, they're intracranially engrafted. And then we use the exact same kind of approach and chemistry that I described to you earlier. And then for a subset of these, we in parallel perform cell surface capture on cell culture models. So it's the same chemistry as here, but it's not a magnetic bead, but a biotin moiety. So you can actually now detect glycoproteins spatially resolved, in particular cell surface proteins. And for the brain cancer, we use patient-derived brain tumor initiating cells. So these are CD133 positive cells from a GBM tumor. It's actually the exact same cells that we're engrafting in these animals. So we have now glycoproteomics data in the PDX as well as in the culture model. And then there's some carefully selected immortalized cell line models for head, neck, lung, and ovarian cancer. And then here's just a couple of data slides. So obviously we just generated this data. This will keep us probably busy for years. So here is a subset of human unique proteins, so only based on human unique peptides. And here in this kind of complicated 3D PCA, you can see they kind of separate by tumor type, and they also kind of separate by engraftment type. So the ovarian cancer, the IP engrafted, they always have the most variability. That engraftment strategy is just the hardest. Here's your intracranially engrafted GBMs, and then your your sub-Q engrafted tumors. So here, separating somewhat by tumor type. You can see it so nicely here. If you look from top, you can see it nicer. And then here's the same strategy with only the mouse unique peptides. So this is now basically looking at the stroma of these PDX models. You can see here, obviously, there's a big separation between mouse cells or stroma cells from the brain, from an IP engraftment, from a sub-Q. If you look a little bit more careful, there's probably some separation of the sub-Q engrafted mouse stromal cells based on tumor type, which is interesting. And then what Minusha is now most interested in is using these data to mine and select surface molecules initially for molecular biology, eventually for the generation of antibodies for immunotherapy. And here's kind of the strategy for mining that she came up with. So there's around 1,300 or so human unique proteins in these 7 different PDX models. First, we mine it for robust expression and high expression in these PDX models. So it needs to be in more than 50% of the PDXs in a higher expression by this intensity that I showed you earlier. Here, obviously, we don't do spatially resolved detection, so we predict surface localization using cell surface genie from the Gundry lab. And then what's most importantly, if you're in the future already thinking about generating some kind of antibody-based therapeutics, you need to kind of mine against expression in normal tissues. We do this kind of in 2 tiers. First, we look at Human Protein Atlas and GTEx, RNA-seq, and proteomics. And more recently, we also looked at cardiac and brain data from the Gundry and Custer lab. There's some very nice proteomics data sets appearing. And then here you can see kind of markers that show this limited expression in normal tissues. And we actually benchmark this and we select that threshold that we're interested in using currently available ADC or CAR-T targets. So these are either FDA-approved ADCs, antibody drug conjugates, or CAR-T cells, or in late-stage clinical trial. You can see this is the total number of ADC or CAR-T targets that are out there with these criteria, and in dark is those that are detected within our PDX database. And so basically 28 ADCs, as I said, that are either FDA approved or in late-stage clinical trial that are detected in this tissue. And then we use their expression in these normal tissue data sets as a threshold where we benchmark, where we basically draw the line. So that gives us around 460 or so targets. And here you can see how they split in these 7 different tumor types. Of course, some of them are detected in all of them, and some of them are uniquely detected in these tumor types. And again, you can look at some kind of emerging ADC targets, and they pop up in the right tumor types in these models. So I think I'm running out of time, so I'll just summarize. So I've shown you here in this partially published, partially ongoing work that we can use PDX models and glycoproteomics to detect potential serum-based biomarkers. As part of this ongoing work, we try to verify previously detected markers, and it looks like one can verify them in independent cohorts. We found this LTBP1 that certainly has longitudinal expression profiles that nicely mimic CA125 ELISA, possibly rising earlier than CA125 ELISA. We expanded this PDX platform to additional tumor types, and we've selected a whole bunch of surface markers for molecular biology through knockout studies in these model system that I've showed you, where we have parallel cell surface capture data. And then obviously the long-term goal, can we use some of them? You have to be highly limited, some of them to raise antibodies for either an ADC or a CAR-T target. And that's it. So here's acknowledgements. Of course, all this work was done by great students and postdocs in the lab. ANKIT high-grade cirrus models. Debbie verified these markers. Minusha extended it. all our collaborators, funding. I thank you for listening, giving me the chance to speak. Happy to take questions later, and I hope I wasn't too long. Thanks.
Okay, thank you, Dr. Kisslinger. That was very interesting to hear about all of your latest applications of glycoprotein profiling and your identification of some potential biomarkers. That all seems like really exciting research, and we'll be able to discuss that at the end. Don't forget, you can enter your questions in the Q&A if you have any questions for Dr. Kisslinger. And next, I will move on to our next speaker. It's my pleasure to introduce Dr. Wilhelm Haas. He's from the Harvard Mass General Research Institute. Dr. Haas's lab utilizes quantitative mass spec-based proteomics to characterize cancer cells and identify potential vulnerabilities. And their technologies focus on protein-protein interactions, high-throughput proteomics, and understanding how drugs alter the cancer proteome. So go ahead.
You are muted, Professor.
Everybody seeing the slides?
Looks great.
Okay, perfect. It's an honor to be here. I want to start thanking the organizers for giving me the opportunity to present some of our work. So my name is Willie Haas. I'm a PI at the Crown Center for Cancer Research at the Mass General Hospital in Boston, and Department of Medicine at Harvard Medical School. We are doing proteomics, mass spectrometry-based proteomics, and the main implication is on just a wide range of questions in cancer research, with some focus on trying to use mass spec to identify cancer vulnerabilities, tumor vulnerabilities in a patient-specific manner. Slow. Okay. This here away on my side. So mapping the cancer nectome to identify cancer vulnerability, that's what I want to talk about today. When we think about how to predict cancer vulnerabilities, we mainly think about the cancer genome. This big DCGA project everybody knows of, where thousands of tumors were sequenced and many, many driver mutations were identified. It's those driver mutations that we mainly use today to predict vulnerabilities of cancers, to predict the best research, the best targeted treatment strategies. It works very well for many mutations. For most of them, it doesn't work at this point. And what we wanted to do here as a goal is we wanted to see, okay, when we move what we're looking at from the genome to a functional level, can we use this as a better predictor for vulnerabilities? And what we use as the functional level, the functional entity of a cell is the endectome, often seen as the link between the genotype and the phenotype. And I will tell you how we access this endectome, cancer endectome, using mass spec, and how we are starting to use this endectome data, the cancer endectome data, to predict vulnerabilities of tumors. I don't know why this doesn't work. It's just slow. Okay, here we go. As I said, we're using mass spectrometry. Very briefly, what is our platform? We're doing bar-coded mass spectrometry. We're using TMT reagents that allows us currently to quantify up to 18 samples simultaneously. We're using an MS3 approach for doing accurate quantification. What needs this approach is some offline fractionation. After pooling our samples, we fractionate them into 12 fractions. Then we quantify some 8,000 plus proteins per samples in 2 hours. This is a throughput, this big issue right now in mass spec. many of you will be familiar with DIA and different technologies. But this is changing currently also in the lab. We're now working on an AI-driven targeted data acquisition method. We really boost this throughput. So I think over the next couple of years, we're going to see a lot of progress there. So how did this whole project start? It started with me starting a lab and us asking the questions if there is anything unique in the cancer proteome to better understand cancer beyond the data that we get from genomics technologies. So in one of the first projects we did is we monitored the proteomes of 41 breast cancer cell lines, and we got very good numbers, and 9,000 proteins in each cell line. And then we looked at the data, and you see here a clustering of this data is a correlation plot. And what we saw here for this breast cancer cell line very well, this clustering in those subtypes, luminal subtypes in red, and the basal subtypes in green, and further subtypes. So this looked very well, very good and encouraging. But what was a little disappointing for us was that the clustering we got was very similar to what we got from the clustering using RNA-seq data of the same cell. So looking nice and giving us what we wanted to see based on what was known, but this very naive, simple approach didn't give us any new information about those cancer cell lines that we could use to further dig in. So we looked a while at this data, and one thing we tried is to turn the clustering around, to not take the proteome data for clustering the cell lines, but take the concentration differences of each of those proteins across all those cell lines to cluster the proteins. And the one thing that we saw is that when two proteins are interacting with each other, this is shown in the upper left here for two proteome subunits, their protein concentrations is closely correlated with each other. See, they're moving this exact same direction across all those cell lines, and every dot is a cell line left to the right, which is interesting. What we found more interesting is that this is a protein-specific thing. We get RNA-seq data from all of the cell lines, and we didn't see this co-regulation of interacting gene products based on the RNA concentration correlation. This is shown here. We see for another two proteasome subunits, we see in a scatter plot a very good correlation on the protein basis, but almost no correlation when we look at the RNA-seq data. So we had something unique for the proteome here. What we could use this, the first attempt, we could use all our proteome cell line data and do a qualitative, a static interactome map. This is shown here. And these are 15,000 interactions across 3,000 proteins that we got through this co-regulation analysis out of those 41 breast cancer cell lines. And roughly half of them were known interactions. The unknown, there was some hot stuff in there because it was membrane-associated proteins and so on. And you see known complexes here shown in different colors and shapes. Okay, so this is something we have extended since then, since it was published in 2017. We now work with this base network from benzalkylin-specific co-regulation network from some 300 data sets and 70,000 interactions across 5,000 proteins. But again, this is just static. What we really wanted, something else. This is just dynamics. We're going to say, okay, what is difference between the interactome of a cancer cell lines and the interactome, let's say, of a normal cell line? And this is how we approach this. So we're mapping what we call interactome dynamics or dysregulations. An example here on the left. So this on the left top is a scatter plot, again, of the protein concentrations of two proteins, DOC1 and DOC2, across a couple of cell lines. Every dot is a cell lines. And you see overall, we see a very good correlation as expected for interacting protein. But there's one outlier. So this one cell line, MDAMB157, there's no correlation between those two proteins. And again, this is protein specific. When we look at the RNA data, we don't see this outlier here. So having a closer look at these cell lines, we found that what made MDAMB157 an outlier is a mutation of one of these two proteins in the cell line. So mutation at DOC2. What we think is happening here is that this mutation in DOC2 somehow interrupts the interaction between these two proteins so that we get a deviation of the canonical correlation. Using this analysis scheme, we can go now to all those 15,000 interactions in each of those different cell lines and check for which proteins carry any dysregulated interactions in the specific cell lines. It's shown here for this specific cell line. You see the base network here, and highlighted in red are those proteins that carry dysregulated interaction in a specific cell line. What was really interesting for us is that those networks of proteins with dysregulated protein-protein interactions, they are highly enriched in cancer cell line specific essential genes, so fitness genes. Okay, so genes, when you knock them out, this specific cancer cell line dies. Okay, so we validated this using 200 cancer cell lines, where we had proteome data on, and also genetic loss of function screen data, genome-wide, from the DepMap project at the Broad. And what you see on these plots that I'm showing here is on the x-axis, you see the fraction of essential genes across all 15,000 mapped by DepMap in this specific cell line. It's relatively stable. It's always about 3% for each of those 200 cell lines. And what you see in the y-axis is the fraction of essential genes after filtering based on our proteomics analysis. In this case, in the first plot on the left, it's the dysregulation, the dysregulated protein network. And you see that this network is highly enriched in those essential genes, moving from 3% to some 30%, and almost 1 in 3 dysreg proteins is a vulnerability of this specific cancer cell line. And we checked if we could get similar information out of looking just for proteins upregulated in specific cell lines, but we don't see an enrichment there for fitness genes. So HER2, breast cancer, is really an exemption there. The same for transcriptome. So here's we had something really exciting, something unique that we only got from the proteome data, a very cost-effective way. Okay, and there's a couple of applications we do in the lab with this model. One of them here shown in the light is to use this for identifying combination treatment targets for synergistic drug pairing. So we can do this. The nice thing about this vulnerability mapping, it's cheap. You only need a proteome map, and you can do proteome map, I don't know, in sub 1 hour most probably. And you can do it on any sample. You can do it on xenografts. You can do it on cell culture model. Anything works. Okay, this is an advantage of using other vulnerability mappings like genetic screens, which are getting really hard when you go in vivo. So for these combination treatment targets, we usually use several models, cell culture, xenograph, which are treated with the drug A, whereby the models show different responses to this drug A. And then we just see what happens in correlation with the drug response to dysregulations of interactions that we induce. And we are specifically interested in those interactions that are dysregulated only upon adding the drugs, because these are a very nice candidate for selecting different additional drugs going exactly for those targets to get a synergistic effects for the drug pairings. Another big project going on in the lab is to really use this technology to map on the broad scale vulnerabilities of cancer types. This project here that's about to be wrapped up is on breast cancer, where we have mapped the proteome and the endectome of a total of more than 1,000 breast tumor and healthy breast tissue samples. And here's the subtype distribution, showing all different subtypes from hormone receptor positive to TNBC to lymph node metastasis, DCIS is a brief form of breast cancer, and normal samples for a big part paired with the invasive samples. This is just a glimpse of PCA separation of tumor and normal samples based on protein levels. I'm not going to go into detail in this project. What I want to go on in the second part of my talk is a follow-up question on this whole analysis. And this was about what are the molecular mechanisms that are underlying or underpinning that the concentrations of interacting proteins are correlated? So we looked at that using, as a model, aneuploid cancer. So aneuploid is gene copy number alterations that affect whole chromosomes, chromosome arms. It's a very common cancer, affects 90% of solid tumors. And in this model that is seen in the top, we're only focusing on 4 chromosomes. One of them is double. And what we think happens is that upon transcription, the differences of numbers of the chromosomes are well refracted, the resulting mRNA molecules. We see 4 greens, but only 2 red, yellow. And we think that same happens through the translation process, that we still end up with 4 green proteins here, but only 2 yellows, 2 reds, 2 blues. But then complexes are formed. It turns out those 4 proteins form one complex. And here it's getting obvious that we have some excess copies here, the 2 green ones. And these partially unfolded proteins can cause proteotoxicity. So we think that something has to happen to get rid of those proteins to protect the cell. It turns out if such a process happens, it would lead to this co-regulation of proteins according to the complexes of the interaction. Okay, and this just shows here that this model is underlined by the data that we have. We see on the left in blue a high correlation between RNA levels and copy number alteration levels that drops when we look for the correlation between protein and copy number correlation. So the correlation between these 2 fields is higher than the correlation between the final protein concentration and the copy number. And what we see on the right is that within a complex, okay, the correlation of members of a complex are very high. This is why co-regulation works. And this drops when we look at the RNA levels instead, where we see a significant drop. So I have, what do you have? Do I have another 5 minutes here? Is this roughly?
That's perfect.
Okay, so I'm gonna hurry up a little bit. But what we did to identify what is behind this co-regulation of proteins, we newly defined what aneuploidy really means. So we said aneuploidy, we watch aneuploidy as the amount of burden per cell to get rid of those excess proteins, okay? So for that, we took all, in this case, 15,000 or 14,000 interactions. And for each of them, we knew that the proteins are co-regulated. We looked at the underlying copy number alterations per cell line. So we knew in the first approximation, they said whatever is in excess there at copy number alteration has to be degraded. Okay, so in the first case, is protein-protein interaction, the copy number alterations, we know the proteins are the same. So in this first approximation, half of A has to go to get this co-regulation. We call this a copy number imbalance. So protein interaction defined copy number imbalance. And we get one value for a cell line, but just averaging all the copy number imbalances across 15,000 reactions for each of those cell lines. And having this one value, okay, which tells us about the burden a certain cancer cell is feeling through aneuploidy and the requirement of getting rid of all of those proteins, we could use this value and analyze it or correlate it with genetic dropout screens and just see, okay, if CNI goes up, are there certain proteins which get more essential for those cells? And we found only 5 proteins here for that, with functionally enriched for those in the ubiquitin proteasome system, so which confirms kind of a hypothesis that it might be protein degradation involved in this process. We focus on an EC ligase, UPF4, to follow up. This is on bioarchives here, Kadiris et al. If you're interested. I'm going to rush through the rest of the slides. Before it's a big protein, it is an N-recognition ubiquitin lysis. So it recognizes its substrates based on the N-terminus of the protein, which might be a good explanation for the high probability security that we think this EC ligase has to really have an effect on adjusting the levels across the entire proteome. We did a couple of follow-up experiments to see if this really holds true with checking different cell lines with high copy number imbalance and low copy number imbalance. And we did in vitro knockouts to show that UBF4 is really essential only for this high copy number imbalances. And we went into xenograft models to also show that the whole thing holds true in vivo. It's very effective to knock out of UBF4 for high CNI cell lines in vivo. We did proteomics to look for what proteins go up in only high CNI cell lines, but not low CNI cell lines upon UBF4 knockdown. And what we see is this whole protein machinery that takes care of ER stress, topologically incorrect proteins, response to unfolded proteins. So again, confirms our hypothesis that this is about unfolded proteins. We're knocking out UBF4, we see an accumulation of unfolded proteins. We think this whole machinery is upregulated to try to get rid of that problem. And then the one thing we really looked at is we thought when we knock out UBF4 in the high CNI cell lines, we expect co-regulation to be inhibited, so the proteins that are in the same complexes are not co-regulated anymore. And at the same time, we expected that the level of proteins is converging with the levels of copy number alterations within those cells. And just in brief, it is we've seen what we want to see here is just that for the high CNI cell lines, we see a higher increase in those dysregulations. So we see a higher decrease in co-regulation upon UBF4 knockdown, shown here in red. Here's a little bit more complicated analysis, but what we show here, without going into details, is that for 7 out of 8 high copy number imbalance cell lines, we saw about UBF4 for those affected proteins that their levels were converging with the underlying copy number alterations. So this leads us to putting, instead of this question mark here, UBF4 as a potentially key player in, I would say, holding collateral damage, okay, that comes from those copy number alterations in control. Of course, aneuploid is very common in cancer. I mentioned this before. It affects 90% of solid tumors. It's connected with metastasis and worse prognosis. We think that UBF4 is a promising target for a wide cancer treatment strategies for aneuploid cancers. Again, this manuscript is in bioRxives. I told you everything from protein co-regulation to how we use this for mapping dysregulated interaction networks, to use these dysregulated interaction networks to identify fault vulnerabilities, and then turn the whole thing around to say, okay, by looking at what makes those co-regulation possible, what is the molecular mechanism underlying this, we identified a very specific vulnerability for aneuploid cancers. The UBF4 project was started by Mina here and is now done by Sambavi in the lab, and Johannes and Rob are mainly working on the dysregulation project. I want to thank all the funding sources, the patients with whom this project wouldn't be possible. I thank you for attention. Looking forward to answering your questions. Thank you.
Thank you so much. That was fantastic. Just really interesting. I really love this idea of functional proteomics, right? Where it's not just quantifying. You have to understand how are they working together and all these other kind of processes that impact that. So I want to encourage anybody in our audience to go ahead and submit questions. I know Amanda and I also have some for ourselves. We have a few minutes here for a Q&A. So I'm just going to start off with Dr. Kisslinger. So I'm just going to kind of combine two into one. So from two of our audience members, they're really interested if you've looked at non-modified peptides, maybe looking at that stoichiometry of N-glycopeptides to unmodified. And then if you're doing any type of normalization or anything like that, as well as do you have healthy controls in your patient cohorts?
Right. So thanks, Nick and Katie. I almost expected Nick will ask me a question like that. So So yeah, at the moment we have not. So we are at this moment in time almost ruthlessly ignoring that glycan structure, which is it makes me sad always, right? I mean, we're developing tools now that would allow us to look at interglycopeptides. And yes, Nick, you're totally right. Occupancy could be a case we could enrich for some funky glycoform that we then can't directly compare to an ELISA, for example, or there may even be information in that glycan structure that's related to disease or disease progression that we're ignoring. Yes, we haven't done this yet. Work in progress. This is a little bit new ground for us. Not like you focus on that. But this is kind of developing new technologies again. So hopefully, maybe in a couple of years, I can show this. And then Katie, also, thanks for your question. So healthy controls, you mean in the context of the first part of the serum marker. So we don't, but we're also, I don't think we need them here, right? So we are basically, if you have longitudinal patient samples, every patient becomes its own control, right? So we're not in the business of trying to do early detection for ovarian cancer. That's a whole different ball game, so to say. Yes, for that you would need well-matched controls for some type of a screening approach. We are interested here, so that's why we don't have them. Can we have markers where the abundance of the marker is correlated with tumor load? Can we compare this to CA-125 to have that baseline? And then obviously our ultimate goal would be, can we have markers that either rise earlier? And there's reasons, there's so maybe long answer, right? So there have been clinical trials that compared. So CA-125 rises 4 months prior to clinical symptoms. There have been clinical trials that try to stratify women with high-grade serous ovarian cancer, treatment based on rising CA-125 versus treatment based on clinical symptoms. And they have suggested that there is no advantage of this early detection. Now you can read the dozens of commentaries on these clinical trials that will inform you that's probably a falsy statement. So our goal is, can we detect this rise earlier? Could we then treat these patients, or in the future, could these patients be treated earlier in recurrence? And would this give you additional kind of opportunities to treat them? So you have a very short window when these women recur before they unfortunately pass away. So you don't have a lot of options of treating them. If that treatment doesn't show you any efficacy, it's probably too late. So if you have multiple options in that at least theoretically, one could hope that this could improve outcome. It wouldn't cure these women, unfortunately, but improve outcomes. So sorry for that long answer, but so we don't have controls.
Thomas, may I
Yes.
ask you, when I look at your first slides where you show your glycopeptides that you have found in the tumor, and then in the sera of the normal and the sera of the PDX, okay? So all of the glycopeptides you found in the sera and the PDX sera, almost all but 10, were also found in the tumors. Is it surprising to you? Is all the glycopeptidome coming from the tumor in this mice? Or is it just because it's super inflamed there and that's why it's just filling up the serum?
I'm not sure. Let me quickly go to this so I understand what you actually, I think I'm not. So you're talking about this Venn diagram, right?
Yeah.
Well, I mean, let me actually share it. They're not real. I mean, let me go to it so I understand your question.
Maybe I also misread it.
I can share actually. So you're talking about this basically, right? Well, I mean, so here you're right? So the vast majority of these glycopeptides that we're picking up are either detected in tumor and serum. That's what you're talking about, right?
Yeah, exactly.
Well, but it could be that the abundance is dramatically different, right? I mean, I'm not. I mean, obviously, right? Also, your tumor tissue still has serum in it as well, right? I mean, you're not completely perfusing this out. So there's probably some of these glycopeptides that we're picking up in tumor tissues are also coming from serum, right? And then ultimately, depends a little bit on abundance ultimately too, right?
Okay. No, get it. Okay. Yeah, you're right. With remaining serum in there, I just wondered if other organs are also shedding off glycopeptides that we should see in the serum, but not in the tumor tissue. But you're right. If this is soaked in blood, the tumor, you will.
There
also
is a
see those.
few, right? There is. I mean, you're absolutely right.
Yeah,
And then, of course, it comes always down again. I mean, even with glyco, right, in serum, you have this dynamic range problem, right? So you'll probably detect way more in tumor than what you do in serum. There still is around 60, 70 that you only detect in serum that you don't pick up in the tissues.
Okay.
Yeah. Yeah, good question. Thanks.
Okay, thanks.
Okay, I think we're a bit over, but we have one more quick question for Dr. Haas.
Okay.
Have you detected any important proteins that formed an outlier in any of your data sets? And if so, how did you adjust your detection criteria to include such proteins?
Okay, an outlier now in the co-regulation analysis. Is this what progress? Yes, these are what we're looking for. Okay, so we're looking for those outliers, and this is what we have studied, and we found that when you look at one cell line, and for this one cell line, you see, okay, for which of those 15,000 or 70,000 interactions that we're looking at right now, for which of those canonical co-regulations do you see an outlier for this specific cell line? Okay, and then when we have looked, usually a couple of hundreds. Okay, depends on what you compare it to. So the cell line based data, it was a little bit awkward because we compared, okay, everything as a base network. It was the network across all cell lines. And then we said, okay, what is different in specific cell lines to an average of all cell lines? Okay, but when we do this for, let's say, the primary tumor tissue, we say, okay, we have a base network that we generate from cell lines. Then we take the tumor data, and then we look in each of those primary tumor tissues for those outliers. Okay, and then we're checking only for outliers that we see in the tumor tissue, enriched in the tumor tissues, but not in the normal tissues. Okay, and these are then those interactions that we think are important to further understand the breast cancer. In principle, vulnerabilities. Obviously, we checked on using cell line data. And those outliers in the cell line data, if you have, let's say, 500 outliers in cell line A, okay, and when you look based on the proteome data, and then you look at the depth map data, so genetic loss of function screens, you will find that out of 3 of those 500 proteins are essential genes for this cell line A. So it's not perfect because you will have passengers in there that we don't really know what they're doing, but they are highly enriched. So you go from some 3% the overall proteome to some 30% of essential genes in your dysregulated network. Okay, this is how we use these outliers. So we're all about outliers. That's all we care about.
Oh, that's awesome. Well, thank you. And thank you both so much for your time and for joining us in US HUPO. We really, really appreciate it, as well as all of our attendees. Just a reminder that all of these US HUPO webinars are available for free for anybody to see. The recording will be put on the US HUPO website. And as always, thank you both so much for supporting our initiative and sharing your amazing science with us.
Thanks for having us. It was fun.
Thanks for having us.